Thanks. One in 3 people in this room will die of cancer. Richard Nixon declared war on cancer, must be 50 years ago now, and that statistic, that 1 in 3, has actually increased in those 50 years. And there are lots of reasons why it's increased. At least part of it is because we've got much better at treating other diseases like heart disease and so on, so people are not dying of those other diseases as much. And it's a really interesting question as to why we've not had the same impact in treating cancer as we have in those other diseases. And I think I'm going to try and take you on a journey through what we understand about cancer now and give you some insights, I hope, into why it's such a difficult condition to treat. And give you some understanding of how it develops, why it occurs in some people and not in other people, and what we might use this information, how we might use this information to better treat people in the future. So I'm going to this is going to be mostly about cancer genetics, the genes that cause cancer. So I'm going to walk you through what a genome is. You would have heard that I spend a lot of time sequencing genomes, and you may or may not know what a genome is, so I'm going to spend some time talking about that. After that, I'm going to tell you a little bit about what cancer is, and why it's related to what a genome is. And then I'm going to take you through 3 little stories. What causes the genetic changes that cancer cells get, how the cancer develops over what sort of time period, what are the kind of changes that are important for cancer, how many are necessary to cause cancer. And then I'm going to finish thinking about some of the ways that we can use this information to help patients with cancer and better improve the outcomes of people with cancer. So, what's a genome? Well, I mean, we all have a sort of understanding about inheritance. You look at your sister or your brother and you think, goodness me, they really are like me. And that's because we inherit DNA, basically, from our parents. And that DNA, the entire DNA that we inherit, is what a genome is. It's that sort of recipe book, if you like, that makes up our cells. And essentially, so if you think about a recipe book, this is the recipe book that all New Zealanders get when they leave home, and this is my copy of said recipe book, and you'll see that's not used perhaps as much as it could have been. But essentially, those recipe books, that they have a series of recipes that make specific things. And so if you want a dessert or a sandwich or whatever, that there'll be a recipe for it. And essentially, our genome is the same thing for our bodies. So everything that you make in your bodies, there's a gene for it. So the tissue in your skin, the collagen that holds your skin together, and that some people inject into their lips to give fuller lips. That collagen. There is a series of genes that make collagen, and basically all of the things that make up our bodies are made of these proteins. And so these genes, they're like the recipes for each of these proteins. And a genome represents the collection of all of those genes that our bodies use. And like a recipe book, individual cells use particular recipes to do things. So your liver cells, they use the genes that are important for making proteins in our blood, for example, and our bone cells, they use genes that are important for making the bone minerals and so on. And so each cell uses a different set of recipes to do its function, basically. Now, Your recipe book that you have sitting on your shelf in your kitchen, that uses a particular alphabet. I guess in your case it uses Danish words to describe the recipes that you interpret and turn into, you know, adding a bit of flour and a bit of eggs and that kind of thing. Our cells in the genome, that recipe is written in DNA. And just like the alphabet that we use in writing, the DNA has an alphabet. And it happens that the DNA alphabet has only got 4 letters, which are A, C, G, and T. And there is a way that our cell interprets that alphabet to make these proteins. And that's something called the genetic code. Now, 4 letters isn't very many. And so in order to turn those 4 letters into the vast and wonderful complexity of a human being, there have to be a lot of letters. And basically our genome consists of about 3 billion of these letters. And they're on strings. They're like a string of letters, hundreds of millions of bases long. And they get folded up. The DNA here, they get folded up into increasingly tight packages into chromosomes. And we have about 22 chromosomes plus 2 sex chromosomes that we inherit, one chromosome of each of the copies from each parent. So that's our genome. And what has really transformed our understanding of cancer biology and indeed many other diseases has been the ability to now analyze that genome. So we can read the genome in much the same way that you can read a recipe book. So as Lars said, I'm working at the Sanger Institute. The Sanger Institute was where the UK made its contribution to the Human Genome Project. And this was a project that ran in the late 90s to sequence the entire human genome. And the UK contributed about 15-20% of that. And that Human Genome Project was a huge worldwide effort to sequence it. And when we did that, I was still at school, but when the scientists at the Sanger and elsewhere were doing this project, you know, it was a very painstaking process. They used initially this kind of technology called slab gels, but most of it was done with this type of technology called capillary sequencing. And essentially, what that allowed them to do was to sequence a few hundred of those bases at a time, and they could do, about 100 or so of these molecules in a day, and then they gradually just turned the handle. It was like a factory. They just churned through these molecules of DNA one by one and built up the sequence of the human genome. And that cost about £1 billion. And sadly, a pound is now worth about €1. So about €1 billion. I don't know what that is in kroner. And it took about 10 years to do. When we did our first cancer genome at the Sanger, this was back in 2009, it cost us about £100,000 to do because there was this new sequencing technology called parallel sequencing. And essentially what you're looking at here, it looks like a starry sky, doesn't it? You see all these kind of white spots on a black background. Well, this isn't a starry sky. It's actually a very blown up picture of what's going on in this new generation of DNA sequencing machines. And essentially, each of those dots is one molecule of DNA being sequenced. And what makes this such an exciting technology is that rather than sequencing 100 or 200 of these at a time, we can sequence 1 billion of them at a time. So each of these dots is basically being sequenced at the same time. And it means that nowadays, we can sequence a whole cancer genome from a patient for about €500, and it takes us less than a week. And that is about the same cost as doing a fancy MRI scan, or the same cost as a pathologist looking down the microscope at your cancer. And what's really exciting about that is that it raises the possibility that we will be able to sequence everybody's cancer as they come through the door and then use it to help understand what would be the best treatment for that person. And that is an amazing possibility, as I'll sort of show you, because there's quite a lot of information in the genome about what caused that cancer, how it developed, and what makes it tick, why it turns into a cancer. So that's what a genome is. Alongside that, we need to know what a cancer is. So a cancer derives essentially from one cell. So our bodies, by the time you're an adult, you have about 40 trillion cells. That's a lot of cells. And each one of those cells has a copy of that original genome. So you start life as a fertilized egg, and that cell, so you start as one cell, and that one cell has to become 40 trillion cells, and every time that cell divides to become another two daughter cells, there's the first cell division, that's that fertilized egg becoming two cells. It has to copy the genome so that each cell gets a copy of that. And then with many cell divisions, you get all of these different types of cells. These are just blood cells, actually. These are all the different types of blood cell that you can get. And you can imagine if you lay all the different organs, there are probably more than 1,000 different types of cells in our body, each doing different things. And each of these has a copy of the genome. And what cancer essentially is it derives from a single one of these cells. So essentially, it's a cell that goes rogue, and it, the cells that we normally have in our body, they kind of function to do things. As I said, your liver cells, they make the proteins in your blood. The neurons, they do the thinking. The brain cells do the thinking for you. Heart cells help pump the blood around the body, etc., etc. So these cells, they have jobs to do, much like a society, really. And what happens, if you like, is that the cancer cell is the criminal of that society. It stops working for the good of society, and it goes and it basically becomes completely selfish and just looks after itself, and it wants to make many millions of copies of itself. And that's essentially what a cancer is. So that growth that you get of a cancer, it all derives from one cell that is just worried about itself, just doing what it needs for itself to get many copies. It tries to take over the body, if you like. And that, what tells that cell to behave in that way are changes in the genes, so what we call a mutation. So we have this copy of the genome, 3 billion bases, and the problem is that's quite a lot of bases. If you had to type out your genome, and you typed it about you typed out a letter every second, it would take you something like 100 years to type out your genome. It's a lot of bases to type out. And every time you copy that genome, every time one cell divides to become two and both of them get a copy of that genome, it has to copy it, and it makes mistakes from time to time. Actually, it's surprising how few mistakes it makes, actually. But it does make occasional mistakes, and those are what we call mutations. So the genome in the daughter cell might be slightly different to the genome that cell was born with. Now, most of those mutations, they don't do anything. The cells just carry on behaving as they do. If they're a neuron, a brain cell, they carry on thinking. If they're a liver cell, they carry on making proteins for your blood. But every so often, that mutation lands in the wrong place in the genome, that genetic change, and that then has a consequence. It's a little bit like this recipe book that you can see I've used a little bit more than the one that I got from my mother, and you can imagine that in copying this, that if you get a change, it's a little bit like a typo. So most of the things that, if you change one of the letters in this recipe book, most of the time it won't have any consequence. You'll still know what it means. Like you'll still say that means an egg or whatever. But every so often, it could radically change the recipe. Like if you imagine that instead of saying teaspoon, it now said tablespoon, and you were putting in salt, that could really damage the recipe. And so it's much the same thing. Every so often, one of these genetic changes, in one of these mutations, lands in the wrong gene in the cell and tells that gene to do something completely different, and something that's bad for the cell and bad for the body. And that's essentially where cancer comes from the accumulation of these particular genetic changes. So, and these mutations, as I say, they occur throughout life. So this is data actually from a normal person that we sequenced the blood from, and what we find is that that even from the first cell division, when this guy was just a fertilized egg, a single cell, we could see that there were genetic changes that were specific to each of the two daughter cells. That only one of them, each one got a mutation, and then we can track down, and essentially we can build a kind of family tree. It's just like the family trees that you drew when you were at school and you were asked to say who you were related to. We can draw a family tree of all of these initial cell divisions in this one person, and we get this sense of the fact that every time these cells divide, they're accumulating one or two genetic changes, and that means that some cells are more related to cells than other cells, etc., etc. And we can build this family tree, but it also means, it's kind of frightening, because it means that no cell in our body has the genome that we were born with. They've all got these little genetic changes. And as I say, thankfully, most of those genetic changes are totally irrelevant, but every so often, the wrong one lands in the wrong place, and that's when cancer can develop. And this is an example of that. This is in a particular type of skin cancer called melanoma, which is the nastiest form of skin cancer. And what you see here, that's one melanoma, and then this is in my field, this is a very famous patient with melanoma, famous as I'll show you later in the talk. But this is what very advanced melanoma can look like. So each one of these lumps that you see here is a deposit of melanoma, and that all of those lumps are the same melanoma, essentially, spread around the body. And each of those derives ultimately from one cell that went bad. And I mean, what it turns out that in this particular person, his melanoma is caused by a genetic change in a gene called BRAF, and this is the sort of data that I spend my life playing with. So I'm going to show you, I'm going to take you through what it is, just so you get a sense of what I do on a day-by-day basis. So essentially, what you're looking at here, you know those little stars in the starry sky that I showed you before, the little white spots, and each one of those molecules was getting sequenced base by base by base by base. That then gets spat out of the machine into this big computer file, and we do lots of quite intensive high performance computing stuff on it. And then this is what comes out. And each of these lines here, the yellow and the blue lines, are individual reads of one of those stars in the starry sky. And what we've done is we've lined all of these up against what we know the genome to be, and we've sequenced the cancer from this patient, and we've sequenced some other tissue, usually blood, from the same person. And then we can see, basically, everywhere there's a red dot is a place where the sequencing read doesn't match what we think it should do. So you can see that in the cancer, there's this one place in the genome where you see all of these red spots lining up. And that same place in the normal tissue, or the blood tissue from this person, there's no sign of that change. So that's a genetic change that's only found in the cancer and isn't present in that person's bloodstream. So we know that that's something that's occurred during the development of that cancer. And it so happens that this particular change, this specific base change, we see in about 30 or 40% of cases of melanoma. So it's a very common change in melanoma. And for lots of reasons, we know that it's one of the major causes of melanoma. This particular genetic change lands in the gene. And the gene, the BRAF gene, it makes a protein that basically tells melanoma cells to divide and ignore everything else that's going on, just make more copies of yourself. And that's why it drives cancer. So that's a sort of illustration of how, if you have a skin cell that doesn't have that genetic change, it probably won't become melanoma. But if it gets that one place, that one spot in the genome, gets that particular genetic change, that's what can tell it to become a melanoma. Now, cancer goes through stages. So that rogue cell, it does start as one cell. And it gets the genetic change that says, start behaving like a cancer, or do something, start to divide. And you get these local growths of cells. So suddenly that cell will become 10 or 20 cells, and it'll kind of begin to behave a bit abnormally. And if that cell, once it becomes 10 or 20 cells or 100 cells or whatever, if it then gets more genetic changes, that if one of those cells then goes and gets a second change that says continue to behave like a cancer or behave more like a cancer, then the cells can start to invade through tissues, and they can start to spread in their nasty little way through tissues. And if they carry on as the cells get more and more in number, of course, there's more and more of a chance that that at least one of those cells will get more genetic damage. And then it can eventually begin to invade through tissues and get into the bloodstream. And then once it gets into the bloodstream, it can land anywhere in the body and basically set up a new colony somewhere else in the body. That's a process that we call metastasis. And it's actually, in the majority of cancers, it's the metastasis that kills people. Usually, what we call the primary tumor, the place where it first grows, we can usually cure that with surgery and radiotherapy. But once it spreads throughout the body, like I showed you in that person with melanoma, when it was all through his body, by that stage, we can't cut all of those things out, basically. And that's why the metastasis is what normally kills people with cancer, unfortunately. So that's a sense of what the genome is, what a cancer is. And so I'm going to show you some data that we've been working with that kind of tells you the first thing, which is what causes those mutations to occur in the first place. So I said that many of them occur when we just copy the DNA, when a cell copies the DNA from one cell to another. But actually the damage, there are things that we can do that increase the rate of these mutations. And of course, if you increase the rate of the damage that you're doing to your genes, then not surprisingly, you can increase the risk that the person will get cancer. So in particular, one of the things, and relevant to the melanoma or the skin cancer example, is that sunlight can damage DNA. You're not at huge risk today, I have to say, of getting much in the way of sunlight damage to your DNA. And what happens is that sunlight, and in particular one type of ultraviolet ray, just happens to be able to hit DNA. If it hits DNA in a particular way, it can damage it. And I don't worry too much about the specifics here, but basically it can damage two bases right next door to one another. And then what happens is that when that damage is there, and the cell's trying to copy that DNA and make a copy of it, it doesn't know what to do with this piece of DNA, and sometimes it puts the wrong things in opposite it, and that's when you get a mutation. So that's how sunlight can cause damage, and that's why when we sequence a skin cancer, this was our very first skin cancer that we sequenced back in 2009, we see a particular pattern of mutations where the C in the DNA gets changed to a T in the DNA, and that's the predominant mutation, as you can see, in skin cancers. All the other types of changes are much less frequent. So that's in skin cancer, and the reason that we see these specific C to T changes is because these bases that get damaged here are Cs. They're what get damaged here, and they get Ts basically replacing them. Another type of thing that you can do to increase the damage to your DNA and give you mutations is smoking. Now, you didn't really expect to come along to a talk on cancer and not hear about smoking, did you? So now the reason that smoking gives you cancer is that the chemicals in cigarette smoke actually bind your DNA. And this is what it looks like. So this is your DNA, and DNA, as you know, is a double helix. And this thing here, that I've circled in blue here, that thing there is a chemical from cigarette smoke bound to DNA. And you can see why it causes mutations, right? Imagine if you were trying to copy this, you wouldn't know what on earth that was, and you could easily make a mistake when you were copying it. And that's essentially what happens, and that's why smoking gives you cancer, because essentially the chemicals in cigarette smoke bind to DNA, damage it, and cause these mutations. And essentially, it's a little bit like, I don't know, do you have a game called, it's not really a game, but do you have a thing called Russian roulette here, where you put bullets into you put one bullet into a gun and you spin the barrel and take a random shot? Essentially, smoking is a little bit like Russian roulette with your genome. Basically, you're firing more bullets, or you're taking more shots at your genome. Most of the time, they will land in random parts of the genome, just like firing a blank won't do you any damage. But every so often, you're going to fire a bullet, and if you fire more times, you will fire more bullets, and eventually, chances are you will hit the wrong part of the genome, and that will give you a push along the way towards cancer. So that's how smoking damages DNA, and you can see this is our very first lung cancer genome, which was done at the same time as the melanoma genome, and you can see that the pattern of mutations is rather different to what we saw in the skin cancer, where in the skin cancer we have these C to T mutations. In lung cancer, we see a much more variable mix of mutations, and the reason it's more variable in the patterns of mutations is because there are lots of chemicals in cigarette smoke that each bind DNA in a different way and cause slightly different patterns of genetic change. And it's a much more complicated picture than we get with sunlight exposure. So now the really interesting thing is that it turns out that there are a whole bunch of things which can cause mutations in cancer, and one of the really fun things that we've been doing over the last 10 years is to try and work out what each of these different things are that cause mutations in our genome. So what we do is we sequence someone's cancer genome, and we can look at the pattern of mutations, much as I showed you for the sunlight and for the tobacco smoke, and because they're all different, and we can separate them into different kind of processes that are causing mutations. So these are the sorts of patterns. So what we're looking at here are individual, what we call signatures of a particular mutational event or things causing mutations. And these signatures, they have very distinct patterns. So basically, I don't particularly want to dwell on the details of what the bars represent, but what I want you to see is that there are kind of 40 different signatures here, and they all look different, right? So there are signatures which are just made up of mostly red types of mutations. There are some made up mostly of blue types and some of green types, etc. Some look very kind of flat and bland like this one. Some are very spiky, and so on. But there are all of these different patterns. And what's interesting is that we have now about 40 or 50 of these different patterns that we see across all of cancer. So we've now sequenced and analyzed in this way several thousands, probably tens of thousands of cancers now, and we're beginning to get a complete catalog of these different things that can cause mutations. And the interesting process that we're in now is trying to work out what causes these things. So obviously, we know the sunlight signature, we know the cigarette smoking signature. It turns out that there are some other things in here which are really quite interesting. So this signature here is caused by a toxin called aflatoxin. And aflatoxin is made by mold that grows on food in sub-Saharan Africa. And aflatoxins, this mold is, when people eat the food that's contaminated with this mold, they get exposure to aflatoxin, and aflatoxin gives this type of mutations. And these people get very high rates of liver cancer as a result of eating this contaminated food. This signature, which is very interesting, is caused by a plant called Aristolochia, and the chemical in Aristolochia is called aristolochic acid. And I cannot tell you how long it took me to learn how to say aristolochic acid without stumbling. This plant, worldwide, there are two major sources of exposure to this plant. One is that it's used as a traditional herbal medicine in China and East Asia. And people who take it as a herbal medicine are giving themselves something that really damages their kidney DNA. And people who have taken it in East Asia and have had and developed kidney cancer as a result of taking it have thousands and thousands of mutations that are caused by this particular chemical. The other source of exposure to aristolochic acid occurs in Balkan states, particularly Romania and regions along the Danube. And here, the exposure is thought to be because this plant grows wild and often grows in wheat fields, and the plant just gets picked up when the wheat is made. And so it's a contaminant you can find the chemical contaminating wheat from those regions in the Balkan states. And so there's very high rates of kidney cancer in some of those regions. And again, when you look at those kidney cancers, you find that they have thousands and thousands of mutations that look exactly like this. Here is another signature, which is also interesting. This signature is caused by chewing tobacco. So amazingly, we can tell from the pattern of mutations in the cancer whether someone chewed tobacco or whether they smoked it. And it's kind of amazing that the same thing taken in different ways leads to slightly different patterns of mutations. And it's all because of the way that the chemicals kind of come out of the tobacco and sit in your cells and bind to your DNA. So you sort of see this rich picture of all of these things which can cause mutations in the genome. And the fascinating thing in many ways about this is that in many of these things, we've no idea what causes them. We just don't know what some of these processes are. There are some really mysterious ones, and there are some that I want to know the cause of before I die. So, because they really bug me. They're really strong, striking signatures. We feel like we ought to know what's causing them, and we've got no idea. And you think it would be useful to know, right? So that's what causes mutations. Then I sort of take you through how cancer develops and over what sort of time frame. Cancer, I sort of talk about it as a sort of orderly, logical process that a cell gets a mutation and it kind of grows, and then it gets another mutation, it grows a bit more, and then another one. But of course, what's happening is actually that this is not an orderly process. Mutations occur randomly. It's a bit like Russian roulette. You're just shooting randomly into your genome with these mutations, and these cells, they're not choosing to get these mutations. They're just happening as the cells go through life. And so you have this kind of complex pattern of changes. And it takes actually probably quite a long time for a cancer to develop. So to sort of illustrate that, I'm going to show you some data from kidney cancer that we've been looking at. In many ways, this is because when I was a child, I wanted to be an archaeologist. like everybody did, right? Really exciting thing to do. But I realized that I was just hopeless at doing any of that kind of, bending over, brushing away little bits of dirt and finding things. I would probably just break everything I found. So this is my sort of trying to be an archaeologist, if you like. And essentially, what happens in kidney cancer is the first genetic change is actually a big chromosomal change, the first important change. And what happens is that, so this is your chromosomes, and you've inherited, this is actually chromosome 3, and this is chromosome 5. And what happens, the first event in kidney cancer is that a bit of chromosome 3 gets lost, and it gets replaced with an extra bit of chromosome 5. That's probably the first thing. You can sort of see it here. This is each of these colors. Each of the chromosomes is a different color depending on what chromosome it is. And you can see this particular one has a bit of 3 and a bit of 5 stuck together. And that's the first key event in kidney cancer development. Now, it so happens that that's quite useful for us, that there's a bit of 5 that gets copied. Because now, if you imagine that as this kidney cell that becomes a kidney cancer, it's been going through life, right? It's just been chugging away, doing its usual thing. And because all cells get mutations as they go through life, it gets mutations as it goes through life. And we know that these mutations in kidney cells occur at a pretty constant rate throughout life, about a mutation every 2 or 3 weeks. And essentially, so what happens is that when this piece of DNA gets copied, and you get an extra version of it in the genome, every mutation that was present on that copy also gets copied and is now present on 2 copies out of the 3 copies of chromosome 5. But then as that kidney cell continues going through life and keeps getting mutations, thereafter any mutation that occurs will only be present on 1 of the 3 copies of 5. So it means that we can use the number of mutations present on 2 copies of chromosome 5 versus the number of mutations present on 1 copy of chromosome 5. And that gives us a rough guide as to when in that person's life this event, the copying of chromosome 5, happened. It's a little bit like carbon dating, for those of you who know what carbon dating is. Basically, you're kind of using the mutations as a clock to tell you when something happened. And it turns out, this is one person's kidney cancer, and all of these mutations here, circled in red, are those that are present on 2 copies of chromosome 5, and then all of these mutations present in blue are found on 1 copy. And you can see that there are many, many more mutations present on 1 copy than there are on 2 copies. And that tells you that this event, this first event in kidney cancer, must have happened pretty early in life. And because we know that these mutations occur like a clock, we can actually estimate when in life that first event happens. And it turns out that it seems to happen in adolescence, when you're a teenager. This first event in kidney cancer is happening so early in life that it's happening when we're growing, essentially. And even though the kidney cancers themselves don't emerge in the patients until they're in the kind of middle-aged to older age group. But the first seeds of kidney cancer are being sown in adolescence, it would seem, that first genetic change. And we think that that first genetic change probably only results in a few hundred cells. So by the time you kind of enter adulthood, there are probably, each of us, probably everybody in this room, will have a few hundred cells that carry that first genetic change of kidney cancer. And then the second change, and then beyond the next few changes, they take a further decade or 2 to accumulate. And then gradually, as the thing begins to expand, it gets more and more aggressive and grows more and more quickly. But those first few changes probably take decades to occur, and probably we're all sitting around carrying the first seeds of kidney cancer in our kidneys. So that begs the question, well, if the first change or 2 take this long, how many changes do you need to get cancer? And we've been working away at this. I don't think we've got a complete answer yet. But this is our sort of best guess at the moment. It's by no means is this complete, but But essentially, we think that most cancers probably have a handful of changes. This is an underestimate. There will be other changes that are important, but it's probably not hundreds of genetic changes that are specifically required to make a cancer. It's probably in the order of a small handful. There do seem like there are some cancers that can be caused by a single genetic change, but they're pretty rare. Most of the adult tumors that we see probably require a handful, something in the order of 5 to 20 mutations to really cause cancer. And it's probably the case that different cancers have different numbers of mutations required for reasons we don't really understand. So, the sort of final bit of this really is, what on earth can we do with all of this information? I mean, I would say that the last decade has been absolutely transformative in our understanding of the basic building blocks of cancer. We understand cancer, the biology of cancer, in a way that we never did 10 years ago. We really know what the genes are that are important for driving cancer. We kind of know how it ticks. We're beginning to understand how it develops. That's quite a long way away from making a difference to patients with cancer. So I'm just going to illustrate some things which I think will happen over the next 10 or 20 years. And don't hold me to account if they don't happen, but this is my prediction. So the first thing is really to follow up on that data. You know, the stuff that I was talking, like that aristolochic acid, the plant that is used as a traditional herbal medicine and is a contaminant of wheat supplies in Romania. It turns out that if you look at most cancers, actually, if you look at the worldwide frequency of these cancers, you find that there's this amazing variation from country to country in the rates of individual cancers. This is what the worldwide distribution of cancer of the esophagus, the gullet, looks like. And you can see that there are places in the world, particularly the east coast of Africa here, where there are very high rates of this type of cancer. And we've got no idea why. They don't have high rates of smoking particularly. There's no, as far as we know, there's no genetic reason for them to, no inherited reason for them to get it. It could be something in their diet. It could be a local virus. It could be some lifestyle, social thing that they do. It could be something in the atmosphere. It could be pollution. We just have no idea. But it's quite possible that whatever it is that's causing that high rate may read out in the genome. We may be able to see a particular pattern of mutations that may give us a clue as to why East Africa has such a high rate of esophageal cancer. So one of the things that we're doing, and this is a project funded by Cancer Research UK in one of these grand challenges where they give £20 million to a team to go away and work on some massive global problem. What we're doing is we've teamed up with a group in France who have collected cancers from all around the world, particularly in some of these hotspots. And we're sequencing cancers from countries where the incidence is very high, and the same cancers from places where it's low, and we're going to compare them, and hopefully we'll find things that we could do something about to help prevent those cancers and to reduce the incidence in those high incidence countries. So that's one thing we can do. The second thing that we can do is to use this information to try and predict what's going to happen to a patient with cancer. Now, the basic thought process here is that these genetic changes fundamentally drive why a cancer behaves like a cancer. It's what causes the cancer. What determines the cell. It tells the cell to behave like a cancer cell. So it turns out, I didn't particularly talk about this, but it turns out that there are lots of different types of genetic change and patterns of genetic change across cancers. So, for example, we have sequenced about 1,500 patients with leukemia. I'll show you, this data comes from that group of patients. So 1,500 patients with leukemia. And we've identified the genes that cause each one of those leukemias. And when we look, there are lots of, because each leukemia will have somewhere between 1 and 5 mutations causing it. When we look at the different combinations of genes that cause that cancer, across the 1500, there are more than 1,000 different combinations of genes that can drive those leukemias. So it's really the case that every person's cancer is unique to that person, to that cancer. And that is kind of frightening, because if you want to design a treatment for cancer, it kind of suggests that you've got a big problem, right? You've got a lot of different types of cancer, a lot of different genetic changes to treat. So in one sense, it's quite scary that there's this much variation in what causes cancer and the particular combinations of genetic changes. But on the other hand, it also explains something that I had wondered about, and many people had wondered about, when we treat patients. So I would see, as I said, I used to treat patients with blood cancer, and I would have, in the same morning in clinic, I'd have two patients with, when I looked down the microscope at their cancer, when I talked to those patients, when I looked at their x-rays, their cancer looked absolutely identical to one another, right? They just looked the same. And yet I would treat one, and I'd treat them both with the same chemotherapy, and one would be cured, and one would die. And what we kind of get the sense of is that actually at least some of that variation in outcome is caused by the fact that the genes are different. In other words, the patient who died had particularly nasty genetic changes, and the patient who we were able to cure had relatively better genetic changes, if you like. Their cancer was more treatable because it had a particular combination of genetic changes. So one of the things that we're doing at the moment is to take all of this genetic information, and we need to do this at big scale. We need thousands of patients with a given tumor type. We map out all of the genes, like those 1,500 patients with leukemia. We map out all of the mutations, and then we know what happens to those patients, and we can build prediction, basically, a black box that can take in this information and can make a prediction about what will happen to a patient that's sitting in front of us in clinic. So basically, you have your patient sitting in front of you in clinic, and you've sequenced that person's genome, and you know everything else about them. You know how old they are and how healthy they are and all the rest of it. And you have a big knowledge bank of, let's say, a few thousand patients with the same leukemia, and in that knowledge bank, you have all of these people that you know what the genetics of their cancer was, and you know what happened to them, you know what treatments they received, and you know whether they lived or died. And basically what we do is we take the patient that's sitting in front of us in clinic, and we compare that patient to the few thousand in our knowledge bank, and we can make a prediction about what will happen to that patient and how likely they are to survive. And we've been building these models mostly for blood cancers to date, and we can actually make pretty accurate prediction. So this is one patient, and we've built a web tool whereby you can enter the information about what genes they have mutated, and then you can calculate what the kind of chance of them being alive at a particular 3 years after treatment starts. So that's one thing. We can predict what will happen to a patient. What's really interesting is that we can go the next step and use this information. We can kind of predict what would happen to the patient if we gave them different treatments. And so in leukemia, which is where we did this initial study, one of the big questions is who should get chemotherapy and who should get a bone marrow transplant. And this is an unsolved question as to who should get what. And this is a particular patient where I'm sort of illustrating the sort of calculation that you could make. And this particular patient is a real patient. He's a 49-year-old man who came in with acute leukemia, and he had this particular set of genetic changes. And what would happen to him at the moment is, on the basis of that particular genetic change of this NPM1 mutation, he would be considered a good risk leukemia, and he would just get chemotherapy. But we can, with all of this information, we can predict what would happen to him, and we would predict that with chemotherapy, he would have a 55% chance of being alive at 3 years, but with a bone marrow transplant, we could improve that to a 69% chance of being alive at 3 years. And so we could use all of this information to change his treatment and to give him a treatment that's better suited to him, more likely to have a benefit. And that is, I think, going to be an exciting use of this kind of information. It requires us to build large knowledge banks. That's where the difficult thing comes. And I was, in my conversations earlier today up at the university, we were talking about how Scandinavia has a particular advantage in being able to build these knowledge banks because you have such a comprehensive healthcare system and you have a culture of people being willing to donate their information and to share their information, consent to their information being used for the public good. This is not such an easy conversation to have in the UK, although it is beginning to start, actually, and we are beginning to think about how we can use this health data to better support the care of future patients that we're seeing. But I think this is going to get a more urgent sort of conversation that we need to have over the next few years, because the promise here is really tremendous, that you could use this sort of information to help decide what treatments we give to patients. And the final thing that we can use this information for about the genes which cause cancer is to design treatments that specifically target the genetic changes. So this is that person, and this is why this person's so famous, this person with melanoma. He was one of the first patients to get a drug that specifically blocked the action of those BRAF mutations. So these drugs, they switch off the BRAF mutation. And this is the same person 3 or 4 months after starting treatment. And you can see that all of those skin lesions that he had have melted away. He's put on a bit more weight and looks a bit more healthy, helped, I think, by the slightly different coloring of the photo, but still he looks much healthier than he did. And he was completely well at this stage. And it shows the sort of ability to use this information to design therapies that are much more targeted and much more effective at treating a particular cancer. Unfortunately, for this particular man, it didn't end all that well, and not long after he was well, his melanoma came roaring back. And what you can see is that everywhere that he had a lump in the original presentation, like this little chain of lymph nodes here, they've come back in exactly the same spot a few months later. And this is one of the problems and one of the reasons that we are having such difficulty in curing cancer, is because it's such a slippery beast and it becomes resistant to so many of the treatments that we use. And it turns out that resistance is also encoded in the genome. So many of the things which allow a cell to evade a drug that we give it are because of mutations in the genome. It comes right back to the same thing that causes cancer. And so what we need to work out now, and we're beginning to do studies on this, is how to combine drugs, much the same way that one way to combat antibiotic resistance is to combine different antibiotics, so that if a bacterium is resistant to one, it will get killed by the other antibiotics. It's the same principle for cancer cells. So if we can use combinations of treatments that basically makes it less likely. We're also beginning to use methods to harness the immune system to attack cancer cells. And that's a really exciting possibility, because you could imagine combining something that directly blocks the genetic effects and then uses the immune system to mop up whatever remains. And so there are lots of really exciting things. The combination therapies in this particular case really radically reduce the time of resistance to emerge. So it's a rapidly changing area, and I think a very exciting time in the next 5-10 years where we begin to use this information to design better treatments and make more effective decisions for our patients. Thank you.